The first step in meiosis is interphase. In the first stage of interphase, the cell replicates its DNA. It is the longest part of the cycle. The second stage of interphase is called nuclear division. In this stage, the chromatids containing DNA are distributed to nuclei as chromosomes in the new cells. The last stage is called cytokinesis. The cytoplasm divides to form the final cells. In interphase, the cells begins to grow and doubles its DNA. DNA synthesis then occurs in preparation for prophase. In prophase, the cell's chromosomes condense and become more tightly coiled and folded. By condensating, the chromosomes are more mobile around the nucleus. Homologous chromosomes are now closely associated in synapses and then trade different segments by crossing over. When mother and father cells pass on traits, their children receive the trait information and store it in their DNA, which then gets copied. Sometimes the child gets all dominant, all recessive, or a mixture of two. In metaphase, the nuclear membrane will disappear and microtubules forming things called spindles, which then form to the spindle apparatus. Spindle fibers attach to one side of each centromere, and both chromosomes attach to the spindles. In anaphase, microtubules of spindles shorten and pull chromosomes towards poles. Each pole has a haploid set with both members of the homologous pair. In telophase, the nuclear membrane reforms around the nuclei. Each nucleus contains two chromatids for each chromosome attached to the centromere, and because of crossing over, the two chromatins are not completely the same. In prophase 2, the nuclear envelope breaks apart and forms a new spindle, just like in the original prophase. In metaphase 2, the spindles bind to both sides of the centromere. In anaphase 2, the spindles contract and chromatids move towards the poles. In telophase 2, the nuclear envelopes form around the chromosomes. And finally, in cytokinesis, all the parts of the new cell are separated and ready to start meiosis again. With some diseases, the chromosomes are the cause. Diseases like color blindness are like the passing of traits and then grow copies. In the picture, notice how the color blindness gene is passed onto another chromosome through the crossover process.